All right. Hello, everyone. My name is Ani Chattaraj. So I am currently a postdoc with Eugene Shaknovich at Harvard. So this year I am playing a different kind of a role. I was at CCAM for 6 years, and I used to be one of the preceptors in this workshop. So it feels kind of strange to assume a different role, but it's at the same time it's good to be back virtually. So basically, I think for the first couple of slides, it will be sort of follow up of what Les just described to you. So the idea of this presentation would be to demonstrate how we can use multiple kinds of tools to answer some biophysical question. And in order to do that, I think it will be good to start from like very beginning. So for example, in the world of biochemical and biophysical simulation, this is a very basic picture that we have a scientific question that we'd like to answer. And since we're dealing with computational tools, I just put simulators, but you can replace simulators with any kinds of tools, including experiments, for example. So let's say we have a question, and in order to get to some answers, we need some kind of tools, right? So in this case, it's a simulators. But interestingly, once we arrive at those answers, depending on what kind of tools you have access to, those answer can lead to more questions, right? So this is a very important step here. So depending on the kind of tools you are dealing with, after getting some kind of answer, that kind of technique or that kind of strategy may give rise to more questions. And of course, those questions can be answered using those tools. So in the language of systems biology, we have a feedback loop here. So it's basically, like to summarize this plot, summarize this cartoon, it's like tools sort of enable this non-linear growth in the knowledge, right? That's like the way you can arrive at answer, and that answer can give rise to more question, and that it's sort of like a non-linear loop there. And another interesting point here that if you use distinct kind of tools, each of the tool can give you distinct kind of perspective. And once you merge them together, once you integrate those pictures, you can get a comprehensive view. So that will be one of the sort of theme of my current talk, that to this would I'll try to show that how we can use different kinds of tools to arrive at some kind of a comprehensive view of a given scientific problem. Another things to remember when we're dealing with modeling is that there's this always this interplay between this quote unquote real world and model world. So we have a problem in the real world, and we sort of abstract that problem and project that problem into this modeling universe or modeling world. And the reason we do that, because this modeling world is very useful, because once we model the real system, we can get things like, what's the mechanism of a process, or what kind of predictions that we can get from some kind of a dynamics of a system. So that's the utility or usefulness of that particular system. So I think it's sort of important to remember that a model is a useful abstraction, and alternative version of the statement is all models are wrong, some are useful. And we should keep that in mind while we are dealing with modeling a system. And I think another interesting thing we should remember that any biological system is inherently multi-scale. So for example, in this plot here on the extreme lower left corner, we are dealing with like atoms and molecules, and in the femtosecond and the picosecond scale. And on the extreme right hand, we have this full-blown organism, which we are dealing with, let's say, like a meter, feet scale, and on the yeah, it could be years, right? Like a human lifespan, for example. So any biological system encompasses this multi-scale system. So in order to answer the questions, that we need to come up with this abstraction, because of course it's almost impossible to find a particular model, a particular tool which can answer all the questions that you have belonging to this different scale, right? So that's why we need this disparate scale sort of give rise to this different kinds of abstraction. And when you talk about abstraction, I'm essentially talking about coarse graining. And coarse graining simply means what are the levels of details that you are willing to get into, right? So let's take like a very toy problem. Let's say we're interested in the binding events of two different proteins. So if if our question is what is the binding interface of those two protein chains, for example, then we need to model the system at an atomic level resolution, right? Because we are dealing with what are the chemical identity of the individual amino acid type that is driving that sort of affinity between the two proteins. So that is sort of like a atomic level resolution. Now, if you're dealing with another kinds of problem, let's say two proteins, blue protein and red protein, they're sort of diffusing in a three-dimensional space, and once they find one another, they will react. If we're dealing with this kind of scale or this kind of resolution, then we would model something like a reaction diffusion model, that what Les just described a few minutes back. So in this case, the concentration of A and B is function of both space and time. So, but here we are not modeling individual proteins with their internal structures and so on and so forth. Here, each protein molecule is a point particle diffusing in space. Now, if we come to another scale altogether, so here what I'm showing is a protein molecule which is a part of a protein-protein interaction network, for example, so which can include like thousands of protein molecules. Here, what we would do instead, that we would say, okay, each of the concentration of each individual molecules is a function of time, so it's evolving only in time. But due to this law of mass action, if we provide some concentration and provide the affinities of the binding, we can sort of get a reasonable approximation of the system's dynamics, right? So these three plots here in this slide, they are there to show you that depending on the problem that you're interested in, or the kinds of question that you have in your mind, you can abstract your system into the arbitrary level of resolution. And there are different tools out there which will help you to solve each of the problem. Okay, so that is sort of like a very like a modeling 101 type of introduction. Now I will get into one specific example where we can actually demonstrate this point. So today I'll be talking about this system called biomolecular condensates. They are also known as phase-separated droplets, liquid-liquid phase separation, or membraneless organelles. They are different names to suggest or indicate the same thing. So the tools that I will be demonstrating today, they are Spring Salad, NFSIM, and the reaction diffusion capability within virtual cell. And I found that Michael sent this slide a couple of days back, which is very useful. So basically, if I try to sort of locate this tool into this interesting tree, they're right here. So here we have a Spring Salad tool. Here we have Banet gene-based model like rule-based simulator, and here we have the spatial deterministic model. And as you can see, you can arrive at the branches depending on this criteria. And once you look at it carefully, you will know that, again, going back to this idea that what kind of feature you want to understand about your system, that will determine what kind of tool you want to use for your simulation. So the first part will be so this is the system sort of introduction that we are interested in this phenomena called phase separated condensate. So, this is a typical textbook picture from a biology textbook, perhaps, that we have in eukaryotic cells, we have this nucleus, and inside this nucleus, we have this nucleolus. And then there is this other kinds of structures that people have discovered over the last 15 years or so. And people collectively refer to them as membraneless organelle or condensates. Okay, so the defining feature of these structures is that they don't have a plasma membrane around them, unlike like Golgi apparatus, mitochondria, or nucleus, which has a defining physical membrane. They don't have a membrane, so that's why we collectively refer to them as membraneless organelle. Okay, so if we zoom in through, we would find that these membraneless organelles are made of different protein molecules, and individual protein molecules have this interesting structure that so these are polymer chains at the end of the day, and along their chains they have this multiple regions, multiple sticky regions, which can stick with other part of the another proteins, for example, right? So that's this idea of multivalency. So there are these multiple domains in these protein molecules which can sort of interact with their binding partners. And due to this multivalent interaction, they can form this sort of network-like structure. And this network eventually sort of phase separates out from the surrounding. So that is sort of the basic idea of phase separation. And this multivalency is one of the driving feature of the phase separation. Okay, so we will try to understand some of the aspects of this condensate biology. Why are they important? It turns out that they are very abundant in cellular systems. So for example, here is a summary that you can see. These are the different examples of condensate. So they can be found in the cytoplasm, they can be found inside the nucleus, and they are also available near the plasma membrane as well, in the cell membrane. So they're important for all kinds of reasons. I will give you one particular example. So this is the cartoonish representation to show that, let's say you are interested in a particular reaction between this yellow balls and the green balls, right? And this sort of light green color, this is a sort of, let's say, another kinds of protein which is mediating this reaction between the yellow and the green. So what's going on here is that you have a condensate made of this light blue color protein, and they sort of recruit this yellow guys and the what's the color, green proteins, and they sort of facilitate the reaction. So this is an example of a selective inclusion. Okay. At the same time, you can also imagine, let's say that you want to prevent a particular reaction. So in this case, they are the red one and the yellow one. So you sort of scavenge all your yellow particle inside the droplet, and you selectively exclude the red particles from your droplet, right? So this would be an example of a selective exclusion rather. So this is like one particular example how you can modulate biochemistry inside living cells by creating this sort of tiny compartments. But importantly, these compartments don't have a membrane around them. That's the defining feature of the structure. So now to give you a real example from a real publication, so this is in this particular set of experiment, the authors are interested in a signaling system, which is important for, like, your immune system and stuff. So for example, in this particular example, we have this protein called LAT, which is a receptor molecule on the cell membrane. So here we are looking at here that these are the two sort of enzymes that are important for the phosphorylation of this receptor molecule. So the LCK is a kinase which mediates the phosphorylation, and CD45 is a phosphatase which inhibits the phosphorylation. So if you look at in this particular plot, what it suggests that once these droplets form, this CD45, which is a phosphatase, they are selectively excluded from the droplet. So, for example, if you draw a line scan, you see a local depletion of the phosphatase here. So this is sort of a real example of this cartoon that I drew here, which is a selective exclusion of the phosphatase. And you can imagine why, because phosphorylation is important for this particular signaling event. So the cell sort of came up with a strategy to exclude the phosphatase, which can prevent this phosphorylation, so that these droplets are stable for some time window. And just to extend this particular system, like for example, initially you have this LAT condensates here, and you add this ATP molecule, which will enable the phosphorylation. And you can see after some point where the droplets form, we have this local production of actin molecule, actin polymerization actually. And then this is sort of a merging of these two color panels. So here we have a droplet in green, we have actin production in red, and you can see they sort of co-localize nicely with each other. So it's sort of suggesting that these droplets are working as some kind of a local reaction chamber to produce some kind of a signaling outcome for cells. So this is an example like what kind of functions that these droplets can perform inside the cells. And as I mentioned, there are multiple other examples out there. So I kind of briefly talked about this. So this idea is that the proteins that form these condensates or droplets, they have this sort of hallmark feature that they are multivalent in nature, meaning they have multiple interaction motif in the sequence, which can engage into their binding partners. And due to this multivalent interaction, they sort of create this three-dimensional network of molecules or chains. and that sort of network will separate out from the solution and create this phase separated droplets. Okay, so this phase separated droplet can be multi-component. So in this particular case, there are two types of protein molecule which are coming together to form this droplet. But in this particular example, there is one type of protein molecule which is sufficient to create the droplet. Okay, so people usually refer to this type of structure as a homotypic interaction because there is only one type of molecules. But this is example of heterotypic condensates because here you have two different types of protein molecules. So the first question we'll be looking at is this idea of what is the concentration threshold of multi-component droplets. So this is a project that I worked with Les and Michael during my PhD, and I will sort of summarize our main finding using the tools called Spring Salad and NFSIM. So to summarize the problem, as I mentioned, there are two types of, so we can broadly classify this structure into two distinct categories. One is called homotypic condensate, another is called heterotypic condensate. So for a single component homotypic condensate, this is sort of the behavior. So let's say you titrate up the concentration of a particular molecule, and after certain threshold concentration, the phase separation will take place. And beyond that point, the dilute phase concentration would no longer change. Okay, so if you titrate the concentration beyond that point, you see the dilute phase concentration sort of remains flat while the droplets are growing in size and number. Okay, so that is the typical behavior from a homotypic sort of a droplet. But for a heterotypic system, the picture is somewhat different. So for example, here what the authors are doing that so this is a multi-component system. So there are many proteins are part of that particular droplet. So the authors tagged one particular component and they're titrating the concentration of that particular component inside living cell. And what they noticed that the dilute phase concentration, although there is a droplet formation, so phase separation has already taken place, but if you titrate up the concentration of this particular molecule here, this particular protein, the dilute phase concentration keeps on increasing as opposed to this quote unquote expected behavior. And this expectation is derived from this sort of homotypic single component system. Okay. So this kind of behavior sort of gave rise to these questions that, okay, how do we think about the concentration threshold or the saturation point for this sort of multi-component system? Okay. So that was the question that we are trying to answer here. And to answer that questions, we took this example of directly from a chemistry textbook, this idea called a solubility product constant or KSP. So the picture is the following. I think most of are already familiar with this idea that let's say you have you're trying to dissolve salt in water. So you're pouring salt into the water. So from our day-to-day experience, we know that initially if you put salt into the water, you stir it, it will dissolve. But beyond some point, you can pour more salt into it, but you will start to see the precipitation at the bottom of the glass, right? You can no longer dissolve the salt beyond certain point. So if I want to translate this picture into a graph, it would look something like this. So initially we have a regime where we pour more salt into the system and there is no precipitation. Almost all the salts are going into the dissolve phase, but then we will hit some kind of a threshold concentration. Beyond that point, we can no longer dissolve any salt, and all the extra salt will appear as a precipitation here, right? So this is a kind of a phase transition because we have a threshold concentration, and below which we have only one phase, which is the dissolve phase. But after that concentration, we have this two phase. One is the dissolve phase, another is this precipitate salt phase here. So we wondered whether we can sort of take this concept, like the solubility product like concept, and apply this kind of concept into this much more complex biomolecular system called phase transition. So again, this is sort of this idea of KSP that if you're dealing with the salt like silver chloride, this would be the expression of this KSP. So for example, this is the concentration of silver cation times concentration of chloride anion. If you're dealing with something like aluminum sulfate, it looks like this because of the stoichiometry and the valency of the salts are different. So for the mixed valence salt, we have a concentration of aluminum raised to the power 2 and concentration of sulfate raised to the power 3. So that's how we define sort of the KSP in a traditional chemistry textbook. And so the idea was, okay, so can we sort of apply this simple chemical concept into a much more complicated biomolecular system? So again, just to reiterate the point that the idea of this project is to to see that whether a KSP-like parameter can define the saturation threshold of a heterotropic condensate. So before we get into the actual solution, I thought it's interesting to mention the point, and I'm sure when Michael talks about the rule-based modeling, he will mention the point that if we're dealing with something simple like A plus B goes to AB, and we know the on rate and off rate, the traditional kinetic models can easily take care of this kind of system because given the concentration of A and B, and you know these rate parameters, you can pretty much determine all the possible species concentration at any given point of time. So writing the OD is pretty straightforward in this particular case. But if you think of something like this, for example, let's say you have two binding sites for A and two binding sites of B, and each of them can interact, you can see that they can form really long polymer chains, and very quickly it becomes untractable by these traditional kinetic schemes. So in order to overcome this problem, people introduce this idea of rule-based modeling, and you will hear a lot of stuff about rule-based modeling, the technical details, but I'm not going into the technical details of it. I'm just going to show that I'm going one particular example where we use rule-based modeling to gain some biophysical insight. So for example, to answer the question that I posed a few slides back, here I'm using two different sort of solver or tools. On the right hand side, we have NFSIM, which is a rule-based modeling framework. So the way to do here is that, again, we are ignoring diffusion here. So this is non-spatial because we are not modeling the diffusion. We are just saying that we have let's say we know the concentration of A4 and B4. These are two different proteins, and we assigned a set of rules which take care of all the possible binding events between those two proteins, and we are assigning the KD of the binding, right? Biochemical affinity of the interaction. So if we know the biochemical affinity of the interaction, if we know the concentration of A and B, we can actually precisely simulate the system to get an interesting idea that what should be this condensation or droplets, what are the concentration threshold and those kind of things. But on the left hand side we have this spring salad solver where we are actually modeling the structure of the molecule. So here again we have four binding sites. So the pink and the orange are the different binding sites, A and B again. But you can see there are these other purple and yellow sites here, which are there to model the flexibility of the overall chain. So they are the sort of the linker sites, and these are the binding sites. And again we are following the binding rule-based framework, but the main distinction between left and right is that here we are accounting for the structure of the molecule as well as they are diffusing in space. Okay. And obviously it comes with the cost that we have to deal with a smaller number of molecules. We cannot really deal with a large system, but with the NFSIM we can deal with a large system, but at the same time we are losing that information that what is the effect of the structural features and those kind of things. So there is always this trade-off, and as I mentioned earlier, that depending on the need of your question, you choose either left or right. And another interesting point to remember, these are all like a stochastic simulation. So you need to run multiple trials to get some kind of average behavior, as opposed to like a solving OD and PD, which are the deterministic type of solver. So we'll be talking about molecular clusters. So when I say molecular cluster, I'm actually talking about this type of picture. So for example, let's say I'm simulating my system using NFSIM in a rule-based fashion. So this is an example of cluster size of 6. So each of the node is my molecule. So each of the edge is a bond between two different molecules. Okay, so that's this idea of a cluster size. So I'll be talking about a cluster size distribution. And when we are talking about a cluster size distribution, what is happening under the hood is essentially we are characterizing these networks of molecules that are present in the system. So this is an example of this two component system A and B, each having some number of binding sites. So let's say at some lower concentration, we have a cluster size distribution here. So x-axis is the cluster size distribution, meaning how many molecules are part of a given cluster, and y-axis is the fraction of total molecules. So this is, we can think of this as a probability of a given molecule being part of that cluster. Okay, so we can read off the fraction of molecule as some kind of a probability. So we have a statistical distribution here. We can draw the mean of the distribution, and the mean of such distribution we call ACO, or average cluster occupancy. So the dotted line sort of indicates that average of this distribution. So at some lower concentration, we have a very lower ACO of the system. That suggests that most of the molecules that are part of the system, they all exist in the form of a monomer, or which is an unconnected molecule. So there is no connection with that molecule with anything else, right? So there is a lot of monomer in the system. So that's why we have a very low ACO in the system. But if we ramp up the concentration of the molecules at a very high concentration of molecules, we have this interesting, like a bimodal bifurcated distribution. And the ACO, as you can see, that which shifted quite a bit. So now my ACO is almost greater than 1,000 molecules. And it sort of suggests that although there are some molecules that remain in the form of monomers and dimers, there is a significant amount of molecules which are part of this huge cluster in the system. So this is an example of phase transition, and this is happening as a function of the concentration of the molecules that are part of the system. So we are essentially quantifying the phase transition in the following manner. By looking at this cluster size distribution, we can tell that whether the system has undergone a phase transition or not. So the left hand side is one phase and the right hand side is two phase. That's why it's a phase transition. Okay. So I think I defined the molecular pair. So we have 4 sites for A, 4 sites for B, and these are the binding rules. And what we're doing here, we start with certain concentration of A and B, and we let them react. And you can see that after a certain point, they sort of converges to a steady state concentration. And once they reach to a steady state, we take the average of that A and B. So in this case, we're looking at free A and free B. So as you can imagine, initially all of them are free because there is no reaction. They start to react and they sort of reaches to this plateauing level here. And once they reaches to the plateau, we take the average of free A and average of free B, and we take the product of this free A and free B, and we call that product as a solubility product. So remember this analogy of the salt example, that in our case, we have this free A times free B, and that's the definition of the solubility product. So in this particular example, the solubility product is 169, okay, because both A and B is 13. We repeat this exercise for a wide variety of concentration. So we see something interesting. So again, on the x-axis, we have a total concentration, and the y-axis, we are plotting the solubility product. So initially, when the concentration is low, if we titrate up the concentration, the solubility product is increasing. But beyond certain point, you see the solubility product sort of reaches to a flat level, right? So beyond this point, although we're increasing more and more molecules into the system, the solubility product is no longer changing. And this is exactly similar to the salt example that I showed you earlier. And on the other side of the thing, so this is solubility product, means this is the free molecules, so meaning it is the monomer concentration. But ACO sort of tells you what's happening to the other arm of your distribution, right? What is this large cluster arm? So as you can see, where the solubility product reaches to this maximal point, we have this increase in this ACO parameter. So this is so if we combine both of these graphs here, what it's suggesting that we have a regime on the extreme left, which is the lower concentration regime, where the solubility product is increasing, but the clustering is almost zero. So that means most of the molecules remain into this sort of dissolved phase. But then we hit this some kind of a threshold. Beyond that point, this clusters starts to grow. That's why the ACO is growing up. But at that regime, the solubility product no longer changes. Okay, so this is our way to model this sort of phase transition boundary here, because here we have a phase transition boundary below which the system shows some kind of behavior, and beyond that point system shows different kind of behavior. So there is some kind of phase transition there. And just to show this one point, that if we take this 100 micromolar point here, highlighted here, and we run multiple trajectories. So we mentioned that these are stochastic simulations. So we need to run many trajectories to get some kind of average behavior. And this is a demonstration of the point that so we run 100 trajectory per single concentration point. So this is this is the statistics of 100 snapshot for this 100 micromolar point. And for each of the trajectory, you can kind of see that this is the bifurcation distribution here. So each of those trajectories will show some population at the lower left and some population at the right hand side. So this is the bifurcation distribution, and if we sort of clump them together, you get some kind of average behavior of your system, which is plotted in this panel here. Okay, so this panel sort of shows you the average of over 100 trials, and these are the individual 100 trials. Okay, so that's the pipeline that we usually follow. And this is just another sort of simulation technique to arrive at the same conclusion. So in the previous method, we kept the concentration of individual molecule fixed. But here what we are doing, we make the box open, quote unquote, because the molecules can come in and molecules can exit. And as a function of these rate parameters, which is the creation and decay rate, by tuning these two rate parameters, we can sort of tune what should be the monomer concentration at a given point. So that particular monomer concentration we call CMC, which stands for clamped monomer concentration. So we are essentially clamping the monomer concentration inside the system, so that at any given point, when you look at the monomer concentration, not the total, but just the monomer, the total monomer concentration should be more or less similar to the CMC. Okay. So if we titrate up this concentration, monomer concentration, we see something interesting, that again, we see some kind of a thresholding behavior here. So when the CMC is 12.9, we see this total concentration reaches to a maximal level and it stays there. But once it crosses this 13 micromolar range, the total concentration sort of monotonically increases. Okay. And again, this sort of tells you that there is some kind of a phase transition going on. And this is this error bars that you see. This is the average over 100 runs. So if we look at individual trajectories, they would look something like this. And one sort of interesting feature here is that right around the phase transition boundary, which is 13 micromolar, the system is very stochastic or noisy, so that you have so much variability in the system. That's why we have this higher sort of error bar here. So to summarize these two plots, on the left-hand side, we have this fixed total concentration where we keep the total concentration fixed. On the right-hand side, we let them change. And we both of the simulations sort of give pointing towards the same point, because if we look at this plot here, the solubility product can only reach to this maximal level of 169. And in this case also, the solubility product is defined by CMCA times CMCB. So this is the 13. Basically, if you take the square of 13, you get 169. So something is changing around that 169 mark here, right? So that's the point. And using two different kinds of simulation, we kind of showed that there is some kind of a phase transition boundary there. And that phase transition boundary can be arrived at using two distinct kind of simulation strategy. Now, we can obviously play with the valency of the molecule. So for example, what happens if we change the valency of the system? And by valency, I mean how many binding sites you have per molecule. So for example, going from A3B3 system to A5B5 system, you can see that your KSP is going down, and as well as the total threshold concentration you need to reach to that saturation level, that is also going down, which makes sense because if you have more valency in your system, more binding sites in your system, you need less amount of molecule to create this sort of cross-link structure. So that's why with higher valency, we have a lower KSP. But this is another interesting example that what happens if we have some kind of a mixed valence system like a A5B3, for example. So for A5B3, the inset sort of shows you the individual concentration of A and B. So where the individual concentrations are changing in all kinds of direction, once we write this SP or the solubility product expression, that solubility product sort of reaches to the plateau. And as I described in the previous slide, once it reaches to the plateau, the cluster size distribution sort of starts showing this bimodal behavior. So this is again a hallmark of phase transition, and that phase transition is arrived at when the solubility product reaches to a maximal level. So this kind of shows the utility of the solubility product. So here we are going to try to compare our simulation with some experimental results. So again, in this particular system, which I described earlier, that all the different components are held fixed. Only one components we are titrating up one component. All the other components are fixed here. So in order to mimic that system in your simulation, we have this sort of three component system, A, B, and C. So what we did, we initially created sort of condensates between B and C, and then we are titrating up A into the system. Okay, so this is a B and C system where the solubility product is maximum. Now we are introducing the A molecule into the system, and we are titrating up A into the system just to mimic this experimental system. And we see something interesting that this is the free A over total A, this is the bound A over total A, and then this is the bound A over free over total. And inside we have this experimental trend from this particular paper. So you can see that our simulation quite nicely captured the behavior that we see in the experiment. So we have this free A concentration increasing as a function of total A. But interestingly, once we take the solubility product of this ABC, this complicated three component system, we see something very interesting behavior that although individual A component is changing, the solubility product sort of reaches to a maximal level. And if we look at this particular system where we are changing A, B, C all at the same time, these are inset are the free concentration of the individual components. Although these A, B, C's, they all change in all kinds of direction, this solubility product again sort of reaches to a constant level, and that sort of defines the phase transition boundary here. So this was an interesting discovery. And of course, we can mimic the same thing using our spring salad simulator, which is the spatial simulator. So here, as I mentioned earlier, that this is the spatial simulation. So here we can actually account for the molecular structure. So for example, we have A plus B, again, A and B being the binding sites of individual molecules. And you can see that this is the end-to-end distance of the individual molecule. So one molecule is slightly longer than the others. We have a longer A and longer B, but the number of binding sites or the valency of individual molecules remain the same, which is 5 here, both 5 pink and 5 green. So this is the similar kind of behavior. So solubility product on the top and ACO at the bottom. This is the clustering tendency. So we see that the solubility product sort of reaches to a maximal level where the ACO starts to sort of increase. So they are sort of pointing towards the same thing that what we learned from NFSIM. But one interesting thing we should notice here that for NFSIM, both A and B, their concentration, they have sort of superimposed concentration because there is not, these are non-spatial simulations, so they cannot distinguish between A and 4. A and B, because both are symmetric in this particular case. But in spring salad, for example, since the structures are slightly different, we have this different behavior between the free A and free B, right? So these are the kind of things that we can learn from while we compare between like different kinds of tools, for example. This is just another sort of example what we can learn from using this spatial simulation. So on the picture on the right, which shows the structure of individual molecules, so we have this parameter called ISD, which stands for inter-site distance. So this is the distance between two successive binding sites, and then the idea is we can keep this molecule flexible, or we can keep this molecule rigid, for example, where they behave as a rigid rod, and it makes a difference. So this two panel shows the left one, the upper one is the flexible molecule and the bottom one is the rigid molecule. And if you notice the y-axis, the rigid molecules has much more higher tendency to form the cluster than the flexible one, because the flexible one, the maximal here is around 35, but here the maximal is around almost 200. So this is a comparison between the rigid molecules versus flexible molecules. Now, what happens if we change this intersite distance? So the intersite distance is that the longer, so we have two different molecules here, two different length actually. So one is longer, one is shorter. But if you take both the molecules of the same length, so now I have same length of A and B. For the flexible is same behavior as we are showing on the left, but for the rigid one, we see something interesting. So the cluster size initially spiked up a little bit, and then it goes down to a very lower level. So what is happening here, we can actually display that into the movie. So this is the cluster size distribution, which is dynamically changing with time. So initially, we'll see there is some cluster populating this right hand side of the thing, which is suggestive that there is a lot of large cluster in the system. But eventually, this dimer peak sort of getting overpopulated, right? So although we started from a lot of molecules being part of the large cluster, eventually all of them become part of this dimer population here. And that is happening because of this complex interplay between this rigidity of the molecule and what's the gap or spacing between two successive binding sites, for example. So these are some of the interesting features that can be probed using the spring salad simulator, which accounts for the spatial geometry of the molecules. So this is just a summary that I already mentioned, this idea that the purpose of this modeling was to understand how can we think of the saturation point or the concentration threshold of this multi-component heterotypic system. And we came up with this solubility product like parameter, which sort of can help us to think about the saturation point. So here the idea is when the solubility product reaches to a maximal, that is sort of the phase transition boundary. Although when we look at the individual components, they can move all over the places. So recently, we also compared our simulation with an experimental result. I'm not going to go into the details, but it's published already, so you can look at that. Just to give a quick summary, this is the experimental quantification of the solubility product of a two-component system. And this is our simulated results, and you can kind of see that qualitatively they match. But obviously there is some dissimilarity between that, and we kind of went into details to explain why that might be in this particular paper. Another thing that I mentioned, that for any stochastic simulation, like rule-based simulation, we need to run multiple trajectories. So we've also sort of combined all those tools into this Python package. So if you are using this tool, you can actually use this package here, which is sort of like an application note, which sort of can do this multi-trajectory analysis automatically for you, and all the results that I showed you, those results can be obtained using this particular package. And a similar kind of thing, I think this is under the process that Michael is currently working on that. There's this Spring Salad Pi, that a similar kind of multi-trajectory analysis can be done for the Spring Salad output as well. So so we are kindly writing it up. Hopefully it will be available soon. So these are some of the like a data analysis tool that are available to analyze this complicated multi-component system. So for the next 5 minutes or so, I will quickly go through this another question that is primarily part of my postdoc, one of the postdoc projects. So the idea is that so in this condensate literature, we always see this multi-droplet picture. So on the left-hand side, we have this typical, again, a picture directly from the kitchen, that if you put oil into the water, eventually the oil phase separates out from the water phase, right? So this is the eventual fate of the system, that we have an oil phase separated by this water phase. But in biological system, we most of the time see a size distribution. Now the question is, why do we see many droplets instead of one large droplet, right? That is the underlying question here. And there are two parts to the question. One question is more physicsy, which asks, okay, what is driving the merger or the coalescence of two different droplets? And the next question is more biology centric, which is, okay, we have a multi-droplet state in our system. Is it functionally more useful, right? So these are the two different parts of the same question. And one hypothesis could be, perhaps this multi-droplet state is a functionally more useful than having this single large counterpart. So for the first part, I'm not covering in today's presentation. It's already published. You can look it up. The bottom line is, this multi-droplet state is not an equilibrium state. It's a sort of a kinetically arrested metastable state. So it's basically, there's an interplay between the kinetics and thermodynamics, and these are all like a dynamically arrested metastable state. But the question that we'll be briefly covering today is this idea that, are they functionally more useful? Because in biology, of course, everything is under this evolutionary pressure. So whatever is functionally more useful, biology will find all kinds of reason to stabilize that particular configuration, right? So the problem statement could be, let's say we have some amount of molecules in our system, how to spatially distribute them so that we achieve the maximal output. That output can be anything, right? So again, we are dealing with this this actin polymerization system. So we have so this is the experimental system we are modeling. We have this bunch of signaling molecules that form this cluster and condensates. And once they form the condensate, they can locally produce actin around the vicinity of the droplets. So here is the quantification that around the cluster we have a high production of actin, but outside the cluster we don't have a lot of actin molecule. And by outside, I mean in the bulk in the solution, not in the vicinity of the cluster. So this is the basic biochemistry that we are modeling. So we have this actin polymerization module here, which can be captured by this simple OD type of equation, that we need some kind of a actin filament, which is a pre-existing filament. This ARP2 molecule sits on top of this filament and initiates another branch process. So that's why we have this, if we write a ordinary differential equation for F actin, the F actin would look something like this. So G squared, because you need 2G, because I'm not going into the detail, but like you need 2G molecule to initiate the process. So there is a G squared here. And then there is a active ARP molecule that is a necessity at this point. And then obviously there is a pre-existing filament. So then there is a F actin here. So the production of F actin depends on F actin itself. So there is the autocatalysis or a positive feedback loop at this particular process. So in order to answer this question here, I'm using sort of a multi-scale approach. So in the middle, we have this virtual cell model, but on the left-hand side, we have again this bead spring type of simulation using Langevin dynamics, and on the right-hand side, we have a much more coarse-grained version of the system. So if we get into the, so here again, I'm not going to spend too much time, but the basic idea is the following, that we have this nephrin molecule which is diffusing in this two-dimensional surface, and it can recruit all the other signaling molecules once they form the cluster. And once they form the cluster, this ARP molecule sort of resides at the periphery of the cluster. Okay, that's the bottom line of this particular simulation. And once we have that understanding, now we can create this sort of model in virtual cell, that we have this sort of solid geometry as a proxy of the condensate. So we are not modeling anything about the condensate. We are just saying this is the condensate can be modeled by this, approximated by this solid geometry. and ARP23 is diffusing at the periphery of the condensate. And this G and F, they can diffuse anywhere in this three-dimensional volume, but they have a difference between, so the G and F has a difference in the diffusion constant. So that's why we have this interesting behavior. So the upper panel is the beginning of the simulation and the lower panel is the end of the simulation. And we can kind of see that the G actin is locally depleted to produce a lot of F actin right at the vicinity of that cluster. Now, interestingly, if we quantify the result, we get that the red one is the effectin concentration at the surface of the droplet, and the bulk is the blue line, and you can see this difference between the surface and the bulk. So this is some kind of a local production of effectin right at the vicinity of the cluster. And again, this difference between the bulk and the surface sort of goes down as you titrate up the diffusion of the effectin. Okay, so this is again a dynamical property or a kinetic property, because diffusion is a kinetic parameter. If something changes as a function of diffusion, this is not a thermodynamic behavior, this is rather a kinetic behavior. So the accumulation of effectin at the vicinity of the cluster is a function of the diffusion of G and F. And this is so that's why the surface enrichment of effectin is a kinetic phenomena. But this is the most interesting, the juicy part of the presentation is that to asking this question that, okay, we have this one large cluster on the left-hand side, and we have the same amount of ARP in the system. What happens if we split this one large cluster into four smaller ones, keeping the ARP count same? Okay, so we have the total number of ARP same in both left and right. Only thing different is the surface to volume ratio, and the total volume is of course again same from left and right. Now it turns out that due to this difference in the diffusion constant between G and F, we again have this local production of F actin. But now, since we have four reaction center instead of one, we actually gain much more F actin. So the blue line is the global level production of F actin, which is almost same as the red line. So the black dotted line is the effectin that we would have got if we have one large cluster in the system. Okay, so that is the black dotted line. So just by creating one large, four smaller cluster, you actually gain this much. And again, this gain on the global scale effectin production is a function of the diffusion constant of G and F. Okay, so that is the sort of interesting summary that by doing this kind of clever type of simulation, we can actually answer questions like which one is functionally more important, the single cluster versus more cluster. And since I'm almost running out of time, I'm just going to quickly summarize this part here. So I'm not going into the details. So this is again a part of the published work. So you can actually look at the paper if you're interested in that. Here we actually asked that we actually utilized different kinds of simulation tool to understand one particular problem. And the one interesting aspect of this work is each of the simulation is informed by another type of simulation. So this is sort of like a multi-scale way of learning about a simulation. I would like to thank, so there are two different population here on the left-hand side. This is my current postdoctoral group. This is my mentor, Eugene Shakhnovich. So I like to thank Eugene and his group. And on the right-hand side, we have a very familiar picture. This is a picture from 2018 virtual cell workshop. So here is Les, here is me, and here is Michael, and the other members of CCAM. So obviously I presented two different work today. One was done while I was at CCAM. Another was done while I'm currently at. So thanks to both my mentors, and yeah, if you have any question, I'll be happy to take them.